I'm going to take you outside of Australia for this pre-lunchtime talk and talk about this very major problem that we are dealing with, have been dealing with in Europe. Blackgrass, the common name, Alopecuris mysoides. It's really our ryegrass. You've heard a lot about ryegrass this morning. This is our equivalent, really. The fields you can see there, the observant among you will just about pick out a wheat crop at the front of that field, but most of what you're looking at is blackgrass. That's about a month before, 3 or 4 weeks before harvest. So, and this isn't a I mean, obviously this is a fairly severe infestation, but this picture was taken in 2014. Many growers in this situation. So we were fortunate enough around about that time to get funding for a large project, multi-institute, interdisciplinary, really trying to understand this blackgrass problem and resistance in blackgrass from the gene to the landscape level, from mechanism to management. And that's some of the story I want to tell you today. In doing so, I should start out by acknowledging this was a big project, many collaborators, and all of these people had some part in what I'm going to talk to you about today. So I think the part of the project that I want to talk about is really, I've said it was about field epidemiology. One of the things we did in this project was set up a big network, a monitoring network, 70 farms, 200 fields, across which we've monitored blackgrass. This network's still active, so it's now going into its 10th year. But I was really interested in these questions about how can we get a bit more information out of some of our efforts to monitor and characterize resistance. And I think one of the really key ways in which we can do that is by collecting field management data in association with these efforts to map and to describe resistance, because then we can get away from asking questions just about how much is where is it, and perhaps start to ask some of the why questions. Why do we see more resistance here than over here? So we have, over the course of this project, been mapping these populations. I'll just tell you a little bit about that in a moment. Collecting seed, characterizing resistance to a whole host of different modes of action, both at the sort of whole plant and the molecular level. But importantly, we've also collected lots of field management histories. So for each of these fields, we have 10 years of field management history prior to the commencement of this survey. So that's information about which herbicides have been used, which rates, crop rotations, tillage practices. And then we've also got the sort of other economic and environmental data. So what that's enabling us to do is really treat each of these 200 fields as a case study in resistance evolution. So just to give you a sense of some of the data that we've been collecting, as I say, we go out and we map populations. So we walk fields and we map the density of blackgrass in those fields. This would be an example of a field with a high density of blackgrass throughout the field. Just giving you a few other examples, blackgrass throughout the field here, but at a more medium density. Another example here would be a field where blackgrass absent from much of that field, but we're starting to see an emerging problem in one area of the field. As I say, what we have is now, for those 200 fields, we have a time series of 10 years of putting together these sorts of maps. So we're able to see what's happening to those populations over time. I'm not going to go into too much detail about showing you all of that data. I just want you to understand the sort of data we've been collecting. And what that enables us to do is build up a sort of a picture of the national and regional patterns of blackgrass infestation in farmers' fields and how that's changing over time, and perhaps what management practices are successful in driving down those populations over time. So, of course, the thing we're very interested in is understanding herbicide resistance in this species. So a huge amount of data summarized here in this one slide. Basically, we looked at levels of resistance to these 3 active ingredients. So 2 ACCAs, group 1 herbicides, 1 ALS, group 2, the mesosulfuron. And I mean, cutting a long story short, we did dose response experiments for all of these herbicides, for all of the populations. But really what I'm showing you here is just at a national level, the level of resistance to these 3 active ingredients in blackgrass populations. And you can see we still have an epidemic of resistance in blackgrass. just to give you a feeling for that, really the mesosulfuron, so the group 2 ALS herbicide was introduced, or it was commercialized in the UK in 2005. And these results are from populations collected in 2014. So we've gone over the course of less than 10 years from a highly effective blackgrass control herbicide that's highly effective for blackgrass control to 75% resistance in the UK population as a whole. 90% for phenoxyprop, and just over 50% for this cycloxidin, the DIM herbicide. So this would be one of these ACCA's herbicides used for grass weed control in broadleaf crops. The phenoxyprop and the mesosulfuron are being used for grass weed control in cereal crops. So again, hopefully that establishes for you we've got a major resistance epidemic in this species in the UK. Okay. I thought there was one more slide here that I've accidentally taken out. But I can explain what we did then, having established the extent of the problem, both in terms of population sizes, but also in terms of the level of resistance. We went back into that management data and we said, okay, what are the major drivers, both in terms of resistance and population size for this blackgrass epidemic? So we had data on crop rotation. We had data on the diversity of crops which had been used. We've got the data on the pesticides, the herbicides which have been used over that time period, tillage practices, and a whole range of other management and agronomic data for each of the fields. And we said, which of that data is predictive of the level of resistance we see in these individual fields? The only thing we found, and again, on the face of it, this is perhaps not too surprising, but the only signal we found was that the intensity of herbicide use in the previous 10 years was the only signal that described the level of resistance in a field. On its own, perhaps that's not so surprising, but we didn't find that any other factors, so wider crop rotations, different tillage practices, had moderated that level of resistance at all. So, and what we also didn't find, so the signal was that it had been the intensity of total herbicide use which was driving resistance. We also didn't find any signal that more diverse herbicide use in that 10-year period prior to measuring resistance in these populations had any impact on reducing levels of resistance. So there was no evidence in our data that herbicide diversity, more modes of action, had been successful in reducing levels of resistance. So I just want to, so then having established levels of whole plant resistance in the population, we wanted to dig in a little further and understand the relative importance of these two major types of resistance. Again, you've heard something about these earlier. So the target site resistance and non-target site resistance, the more sort of metabolism-based resistance. So, and for the purposes of the questions we were asking, I really make a distinction there between specialist and generalist resistance, because target site resistance, it's very specific to a single mode of action. It gives a resistance phenotype which just gives resistance to that single mode of action. Whereas in some cases, this metabolic resistance gives a more broad spectrum of resistance. So you can have a single mechanism of metabolic resistance which will give resistance across different modes of action. So we make a distinction therefore between a sort of specialist and a generalist type of resistance. And we wanted to address the question, given that dichotomy between these two types of resistance, could in fact herbicide diversity and mixtures, so a longer history of using diverse mixtures or rotations of modes of action, could that preferentially select for this non-target site or generalist type of resistance? Okay, because you could see a way in which perhaps that might account for the fact that we didn't see any signal in our data that those practices were reducing selection for resistance. So what we did, and again, I'm not going to go into too much of the detail here, but what we were able to do was, in all of our populations, establish the frequency of target site resistance. So you see some pie graphs there just showing the frequency of the different known mutations amongst our populations. And overall, at a national level, we're able to establish that the frequency of ALS mutations in those populations is 0.25, so 25%, and the frequency of the ACCA's mutations is 0.43. Okay, so now we've done lots of dose response. We know the level of resistance at the whole plant level from glasshouse work in all of these populations. We now know the level of target site resistance in all these populations through the sequencing the target genes. So we're just able to ask the question, okay, does target site resistance explain all of the resistance we observe? And if we see a one-to-one relationship between the frequency of survival in a population on the y-axis and the frequency of those target site mutations, then that would seem to suggest to us that the resistance we see is explained solely by target site mutations. So this is looking at that relationship for the DIM herbicide, cycloxydim, and of course there's some noise, but we do see that one-to-one relationship. And that's telling us that in this case, when we're looking at DIM resistance to these DIM herbicides, we can explain all of the resistance we see as target site resistance. But when we look at the other two herbicides in our survey, we see that there's actually more resistance present in those populations than we can account for by the frequency of target site resistance. There's something else going on. And of course, we would suspect that that something else is non-target site resistance. So again, I'm sort of cutting a long story short. Through work with some of our collaborators, and the person on the left there is Rob Edwards, and so some of this work was done in Rob's lab, or certainly the discovery of this protein, this GSTF1, as a very good marker for non-target site resistance in populations. So Rob's group did a huge amount of work and established this AMGSTF1 gene or protein as a good marker for non-target site resistance. And what it gives us is then a way, as we did previously with the target site resistance, a way of looking at the phenotypics, so the whole plant level of resistance, and saying, how much of that resistance we see in plants is due to non-target site resistance, or to the level of this marker. And what we see in green, reassuringly, because we've already previously seen that non-target site resistance was not involved, we didn't think it was involved in DIM resistance, and we see there's no relationship there with the green line, but we have confirmed for both the ALS in red and the FOP herbicide in blue that there's a strong positive relationship with the level of that marker, that protein, and the level of resistance in the populations. So if we bring all of that together, what we see is in the UK population, for ALS resistance, the resistance we observe is due to the coexistence of both of these mechanisms. It's the same for FOP, and for the DIM resistance, it's just due to target site resistance. Now really the sort of where we wanted to go with that next was then to come back to the management data and say, have different different herbicide use patterns preferentially selected, or do they have different impacts on that specialist and generalist type of resistance? Because what I would argue is most of when we think about and when we're modelling the impacts of mixtures and diversity on resistance, we're often doing it with an assumption of this target site resistance, this very specialist type of resistance. So what you're looking at here on the x-axis, we've gone into the historical management data, and we've said for each of these 200 populations, how much herbicide diversity have they been exposed to over that 10-year period before we collected the populations. So on the left, the population sort of on the left of that axis would have not had much herbicide diversity historically. Those on the right, we're moving to more and more diversity of herbicide use over that 10 years. And what you hopefully clearly see for each of the three herbicides, and on the y-axis we're looking at the frequency of target site resistance. So what you see for each of those three herbicides is a negative relationship. So the theory holds when we're dealing with target site resistance. More diversity, more rotations, more mixtures, and we've selected against, or the lower frequency of this target site resistance. But then, when we switch it round and we say, how does the historical diversity impact the level of the non-target site resistance, we see the opposite relationship. So we see that historical diversity of mode of action use has actually preferentially selected for the non-target site resistance. So what I should say is that this, we found an interesting result. We have to interpret it with caution, because I'm not here to say that herbicide mixtures and herbicide diversity doesn't work for resistance management. I think it does in some circumstances, but I think what this result told us is it's not necessarily always true. It's not necessarily generalizable. In some instances, I think we have to be aware that just using more and more herbicides is just pushing populations in a certain direction towards evolving more of this non-target site resistance. So that's, perhaps a little controversial, which is why I think we need to sort of think carefully about how widely we can apply that. But my argument would be that we haven't sort of thought about it enough to this point in time. So I think I've got 5 minutes left, so I know it's near to lunch, but one of the other things I've often thought would be really good, and AHRI and others were talking about it earlier, was can we predict the risk of resistance before it evolves? That would be a really valuable thing to be able to do. In the UK, we don't have glyphosate resistance in blackgrass, but we have many of the sort of danger signs that might suggest that we're pushing our populations towards glyphosate resistance. So what we did, so we wanted to sort of test this model that, I would argue that, in all populations, there's some variation for sensitivity to herbicides, even before we've selected for with any herbicides. But hopefully, and normally, the field dose is beyond that level of variation. So we control the population. There's variation in there, but it's not really important if the field dose is beyond that level of. But I think what happens as we start to select with herbicides is we shift that distribution over time. And so in this case, we might say that we've got evolving resistance. The field dose is still highly effective, but there's a small part of that population which we're not controlling. We don't see that as a field failure, but it would be really useful if we could pick it up as a signal in populations before they get to this stage. So that's what we attempted to do. We took all of our populations. 540 grams is the UK use rate for glyphosate to control blackgrass. See the scale on the right there. And hopefully what you can see is our populations are largely fairly highly susceptible to the field rate of glyphosate in the UK. But we go to 3/4 of that field rate, so not much of a drop off at all. And we're starting to come into a lot of, we're seeing in many of our populations 30, 40, 50% survival. Okay? It's not field resistance, but I think you'll agree there's a notable signal there. So we did some more work. This just sort of, the plots show that we do have surviving plants at a normal field rate. We did work to establish that difference in sensitivity is heritable, so there is a genetic basis to these differences in our populations. And we're also able to show that we can shift that dose response from one year to another. And again, there's lots of information on here. The only bit what we've done here is go back into that management data. And as we did before, we've said, okay, we've got variation in these populations. What management data is explaining that variation? And the only thing that came out as really significant was the history of glyphosate use. So what it's telling us is the least sensitive populations are the ones which have had the most historical use of glyphosate. So it's a signal, I would say, that we're pushing our UK populations towards glyphosate resistance, and we should use it as an early warning signal. So, excuse me, I've got, towards the end of this project, we were also fortunate enough to work with these guys in the US who've generated a really good reference genome for us for blackgrass, and that's able. I'm going to quickly go back to the non-target site resistance story, because with the reference genome, we're able to start asking questions about the genomic basis of that non-target site resistance. So again, I'm really cutting a very long story short, and I'm nearly finished, Paul. What we did was look for the genetic basis of this non-target site resistance in two highly non-target site resistant populations. And what you see for the two populations is just the areas of the genome which seem to be associated with that resistance. So if we quickly go through, the point I want to make here is we've just looked at two populations, and actually the genetic basis of this resistance in those two populations, they look the same, they're resistant to the same modes of action, but actually the genetic basis is very different. So it's complicated, and actually, I suspect if we look in other populations, we'll see the same sort of thing. And again, I think that tells us that plants have a number of ways or a number of mechanisms available to evolve this non-target site resistance. So I have 2 slides and then a conclusion. I think we're going to talk a bit more about, I wanted to just set this, I think in the discussion this afternoon, I'm going to be talking a little bit more about the sort of context of pesticide use in Europe and the context against which the story I've just told you is unfolding. We're losing pesticides. We have these targets to reduce pesticide use further, but I'm going to not dwell on those now. That's just, as I say, to sort of set this in the wider context. And just to summarize, so what we have a resistance epidemic in this species. That's not unfamiliar to anyone in this room. It's a very similar story to others have told from Australia. I guess that's telling me it's time to, let me see if I can do it without the slides. So we have the resistance epidemic. It's due to the coexistence of these two mechanisms. Is it on the, it might just be, Yep. We've got some signal showing that herbicide diversity does preferentially select for non-target site resistance. We need to interpret that with caution. I'm not here to say that mixtures diversity doesn't work. Evidence for creeping resistance to glyphosate. Genomic basis of that non-target site resistance was quite different between different populations. Yeah. And again, perhaps I'm being a little bit controversial here, or not controversial, I don't think, but I think what we're doing generally in many parts of the world is sort of forcing or evolving a resistance syndrome in weeds, I think it's particularly the case in weeds because of this non-target site resistance. I think we're selecting for general patterns of increased resistance because of upregulation of a whole host of these sort of defense and stress related families. And I just sort of finished it by saying this is happening against this backdrop of increasingly restricted legislation in Europe. And thank you for staying into the lunch break.
Thanks very much, Paul. Fascinating work that you and a very large team of people have clearly done. We are running behind, but we're going to make up a little bit of time this afternoon, and I have no doubt there will be some questions for Paul, so we'll spend a few minutes before we break for lunch. AHRI.
Yeah, it's a great talk, Paul. I'm just wondering if there's any cross-resistance between the herbicides, right? Because that would obviously favour what you're proposing here.
Yes, there is. I mean, there's particularly between those two modes of action that we look at, there is documented cross-resistance between them. And I mean, that's why we set out to address that question, because it seemed like you could at least, you could see a situation where the diversity might be pushing them towards that mechanism.
And my second question was, what's the level of gene flow in the species?
High, yes. I mean, that's. We've done some work amongst all of those populations. We've looked at population structure, and we find that it's, there's a lot of mixing amongst these populations. There's not a lot of population structure in the UK population. Having said that, there's other interesting populations, there's other interesting studies, particularly in Germany, where they've looked at neighbouring fields and looked at the mutations. I think, was it you, AHRI, was talking, or someone maybe today was talking about the fact that, some studies in Germany have shown that actually the mutations underpinning resistance in neighbouring fields are actually quite different. So in that sense, that's sort of going against what I just said. It's suggesting that sometimes the evolution of resistance is happening independently, even in neighbouring fields.
Thanks, Paul. Really interesting presentation. Do you think the types of modes of action or types of products in that diversity mix could influence whether it's impacting the non-target site type theory? And did you look at that at all in terms of differences there in products used?
So the simple answer is yes, I do. I think that if you wanted to conduct a study to look at what we found there, that can these mixtures and diversity select for, can they push populations to non-target site resistance, you would do it probably between ACCAs and ALS, and that's the two, because we know there's these cross-resistance mechanisms. So I think it probably doesn't. I think there are definitely mixtures where this wouldn't occur. We haven't looked at that because I guess blackgrass was our case study. But I do think, when I look at research happening, for example, in North America, where they've got Palmer amaranth, which is now resistant to 8, 9, 10 modes of action. I do think there's evidence in those populations that there is, whilst there may be some components of the resistance which is very specialist and it's due to mutations which give resistance to specific modes of action, there's also, I believe, and I think I've seen evidence of a sort of general upregulation in these non-target site resistance mechanisms as well.
All right. I think that's it. Thanks, Paul.
All right. Thank you, Paul.